I'm Dr. Lori Snyder from Kingston University. Complete genome sequences reveal duplications in bacterial chromosomes. Bacterial genomes are dynamic. Bacterial genome sequences can change, and they can change due to mutations and rearrangements. This can include duplications, deletions, transpositions, and acquisitions, whether that's acquisition of DNA from other species or acquisition of DNA from neighboring cells that are in the same microcolonies. There's lots of dynamic reasons that the genome of a bacterial cell can change, and these changes can impact the coding and the non-coding regions of the chromosome. It is therefore important to remember that a draft bacterial genome sequence, that data is missing a lot of the context of the chromosome. The species that I work on, Neisseria species, that includes Neisseria meningitidis, Neisseria gonorrhea, and a whole host of non-pathogenic Neisseria species, we have known for quite some time that there's a lot of things that change within these genome sequences. So from the first Neisseria genome sequence data, the first two Neisseria genome sequences were of Neisseria meningitidis, and these were published in 2000. One of them was strain MC58, and that was discovered during the assembly of the strain MC58 data from back then being Sanger sequence data. As that was being assembled and people were making sure that this was assembled correctly, it was discovered that some of those contigs were actually duplications. There was a 32 kb duplication present in the chromosome. So this was identified during assembly. There was a lot of more detailed laboratory work, some long PCRs to actually show robustly that this was a big duplication in the chromosome. 32 kb is quite a large sequence to have been duplicated. And there's actually been some subsequent sequencing of a very closely related strain to MC58 that shows that that doesn't have a duplication. So there are strains that have a single copy of this region and strains that have a duplicated copy of this region in their chromosome. So we know both from this story and from lots of other stories of bacterial genome sequencing that these contigs can be misassemblies when there are identical or nearly identical sequences in the genome. And we quite often see this when we're looking at the ribosomal RNA sequences. So ribosomal RNA loci are present in multiple copies in a bacterial chromosome, and they tend to be the last things when you're assembling. So you find you've got one contig that's the ribosomal RNA loci, but you know that they're present 2, 3, or more times in the chromosome. And they've just been put together by the assembly software into one contig that is the ribosomal RNA loci because they're pretty much 100% identical. So moving on, my group did some investigation of the Neisseria gonorrhea complete genome sequences. So at the time, there were 3 complete gonococcal genome sequences, and those are shown there in a mauve whole genome alignment. So we've linearized the chromosomes and aligned them. And this mauve program will find regions of similarity and give you blocks of color for those regions that are similar to one another. And you can see actually the beginning and the ends, those are similar to each other. They're not the same color, but we've numbered them. So you can see that region 1 and region 1 are both the beginning and the end of the chromosome. And as we go on through, we've got region 1, region 2, region 3, and for the first 2 genomes that are shown there, the top one and the middle one, it goes 1, 2, 3, but in the bottom genome sequence, region 2 isn't there. It's actually elsewhere in the chromosome. Now in the top 2, flanking region 2, there are IS1106 elements. So these are insertion sequence elements, and that looks to be what's happened with region 2 is that IS element flanking that region 2 means that region 2 is in a different location in the bottom strain, FA1090, and it's actually inverted. It's on the other strand of the chromosome. When we look then at region 4, which is shown there in purple, we see that whole region has inverted. So that's what it means by being below the center line as shown in the mauve visualization. And so region 4 is in the same relative location, but has inverted in one of the strains. So relative to one another, there's been an inversion in one of the strains versus the other of two of the strains. This region 4 is also where we see the gonococcal genetic island. So this island is present in one of the strains on the top, abbreviated there GGI. We know this island encodes a type 4 secretion system, which is able to pop out of the chromosome as an extra chromosomal circle. So we've shown this in Neisseria meningitidis and in Neisseria gonorrhea. It comes out of the chromosome transiently, probably as an extra chromosomal circle, but most of the time it's seen in the chromosome. And this is actually also able to self-mobilize because of the mechanism that it makes as a type 4 secretion system. And then there are other aspects of the chromosome that mean that there's rearrangements that have happened, mediated by things like repeats flanking that region 4, but also prophage elements that have mediated some rearrangements between the different gonococcal strains that we can see here. This is a close-up of what's happened with that region 2, where it's moved in FA1090, and then the sequence differences between NCCP 11945, where region 2 is between region 1 and region 3, and FA1090, where it's just completely moved somewhere else by virtue of the IS elements that flank it. And this is region 4, that block 4 that has just completely inverted due to some repeat regions that flank region 4, which in strain NCCP 11945 does include the gonococcal genetic island, but it's not that island that's having a role in the inversion. It's these flanking repeats. So there's a combination event due to the flanking repeats that mean it's inverted in some strains versus others. So looking at all of this information together, doing some comparative genomics, using these complete genome sequences that we have, we're able to come up with a model of what these genomes might have looked like before the rearrangements occurred when you compare two strains and try to trace back what happened and how these things have moved around. But of course, most of the genome sequences that we're looking at aren't complete circular chromosomes. As of 2020, for the Neisseria species, there are 159 complete pathogen genome sequences and only 29 complete non-pathogen genome sequences. So therefore, there's 188 Neisseria species circular chromosomes. So far more than when we did our analysis, and there were only 3 gonococcal genome sequences that were complete circles. But in contrast, there's 2,854 incomplete draft genome sequences as a result of short-read technologies. So it's very rapid, very quick to just produce a draft genome sequence and get data that is necessary and useful for many applications. This has been particularly important for investigating antimicrobial resistance, which is, of course, a big problem, particularly for Neisseria gonorrhea, where it has become resistant to the classes of antibiotics and the antibiotics that we're using clinically against gonorrhea to fight those infections. However, some information will be missed due to the lack of complete chromosomal context. So not being able to see the whole of the chromosome and how those various contexts fit together, how many copies there are of the various genes, where all the regulatory regions fit together, and the whole of the chromosome as one piece. We started an investigation quite some time ago now of non-pathogenic Neisseria. These were isolated from the throat swabs taken from university students, and we expected that we might recover some Neisseria meningitidis, which is why we initiated the study. But in the end, none were isolated. Instead, we isolated 4 non-pathogenic Neisseria species. And as it so happens, 2 of these were isolated from one student, and 2 were isolated from another student. So what we actually have are 2 Neisseria species that were co-isolated from one host, and 2 non-pathogenic Neisseria species that were co-isolated from another host. So quite interesting isolates that we've been able to obtain. These were then subjected to microbiological investigation for species identification, and they were subjected to genome sequencing for further investigation. Identification of the isolates first began in the lab. None of them were hemolytic, so therefore they aren't Neisseria anemoralis. Nitrate reduction tests were done, and they were negative for all 4 isolates, which ruled out a whole host of other commensal species. All 4 of them grew at 35 degrees C, which according to our API NH criteria for using our API strips, that meant that they weren't Neisseria gonorrhea. Now, there's various things in the literature about the types of strains of gonococcus, but we also had other evidence that these weren't gonococci. Given all the criteria for the API NH strips that we're using for identification of our isolates, we knew that one of the isolates, RH3002V2F, was identified as Neisseria cinerea. The other 3 isolates, however, we can only identify them as being Neisseria species. So we knew from our initial results using gram stains and some other identifications that we thought they were Neisseria species. Well, this confirmed, yes, they're Neisseria species, but it couldn't come down to a definitive species identification just from that information. But all hope wasn't lost. We had the genome sequence data, and we took it further there. So we used Illumina MiSeq to start with, and we got that from Microbes NG. So we sent away to have the sequencing done at Microbes NG, and this is how the data came back. So these sizes of genomes are in line with what we would expect from Neisserial genomes. They're about 2 megabases, and the contigs assembly wasn't too bad, so it's not hundreds of contigs. It's a good manageable number of contigs, so we're quite happy with the data that came back. Species identification from the sequence data, we first started by looking at the 16S ribosomal RNA using the NCBI tool. Some species were identified there. And then we looked at the ribosomal MLST using the pub-MLST tool. And some species were identified there. And you can see there's some overlaps. Now, something to keep in mind, Neisseria perflava and Neisseria subflava, they've actually been combined into one, so they're now all called Neisseria subflava. So actually the 2 results there for the first isolate, KU 1003-01, they actually do agree on that final subflava perflava. Okay. We then also looked at some of the other sequence features. So DNA uptake sequence variants can give you an indication of what sort of species you're looking at as well. So Neisseria are naturally competent for transformation. They will take up naked DNA from their environment and use it to either repair things in their chromosome or acquire new DNA. This is a 12-base DNA sequence, and although Neisseria can take up DNA from any of the Neisseria species, there are some slight dialects that vary by species. And so we use this to give us an indication of which species we might be looking at and add to the body of evidence of species. And we found that the DUS1, which is most often seen in subflava flavescens and elongata Neisseria, was found in 3 of our isolates. And the classic DUS, which is most often found in Neisseria cinerea, which that isolate had already been identified as cinerea from the microbiological tests, was found in that isolate. So this was all adding to our body of evidence for these isolates. Lastly, we did a complete genome sequence alignment using the contigs from the sequencing and complete genome sequences from pub-MLST. And this is the splits tree analysis, and you can see that 3 of the isolates align on the tree with subflava, and our isolate that was identified as Neisseria cinerea does match up with cinerea on the phylogenetic tree. So this gave us great confidence, along with the other data, that we have 3 isolates that are Neisseria subflava and 1 that is Neisseria cinerea. So we started looking at the data, and what we found first were there were some regions of difference between our 4 isolates. So we wanted to investigate those regions of difference. And in one of those regions of difference, we found something exciting. We found a type 6 secretion system. And this was actually the first type 6 secretion system fully identified in a Neisseria species. So that was very exciting for us because it was the first one completely identified, and we actually worked with Sarah Coulthurst's group to make sure that we got the complete system there, and that we were using all the correct terminology for looking at this type 6 secretion system. Now, these are interesting systems. If you're familiar with kind of the type 3 secretion system, which has been known longer than the type 6, that's a needle complex that the bacteria use to attack host cells. Yes, the type 6 can be used to attack host cells, but really it looks as though these type 6s have evolved to attack competitor bacterial cells. So they do have some toxins that will attack host cells and other eukaryotic cells like fungi, but primarily they look to be there to attack competitor bacterial cells in their niche to give them an advantage. And evolutionarily, what we see is that this looks like a bacteriophage. The bacteria have taken in bacteriophage, flipped it around the other way, and are using it as a weapon to stab and attack competitor bacterial cells and inject them with toxins. And these effector toxin proteins then are in the competitor cells, and the bacteria that have the type 6 secretion system make an immunity protein to stop themselves from dying from their own toxins. So we found the whole mechanism for the type 6 secretion system encoded in the genome sequences that we had sequenced, actually 3 of the 4 isolates. But first, primarily, we found it in one of the isolates and started investigating that in great detail. And one of the things that we knew we needed to identify was the VGRG protein. So that's the purple protein that sits up at the top of the spiky tip of the type 6 secretion system. And it needs to have at least one, but quite often they'll have several of these. And so my PhD student was working on this, and he said, I'm having some trouble working out how many of these are actually sat at the top of the type 6 secretion system. And that's because the genome sequence is still in so many contigs. Would it be possible, could we please get a min-ion sequence of this isolate so that we can have a complete genome sequence? And that was about all the excuse I needed. I've been coming to these conferences for years and watching the Oxford nanopore technology develop. And I did my postdoc under Mark Palin with Nick Lohman also as a postdoc. And I've been talking with Nick Lohman about how the nanopore sequencing was developing. And I knew Microbes NG was now offering the min-ion sequencing as a service. So we sent away our strain to have the min-ion sequence done, and then we received our sequence back as a complete bacterial circular genome. So we're also excited to get this back as one complete contig, one contig of the bacterial sequence, one circle. We actually received 4 contigs, one of which is the chromosome, and then we've got 3 other little contigs. So the chromosome comes up as 2,376,255 bases with a GC of 49%, totally in line with what we would expect for Neisseria subflava. The GC skew is consistent with what we'd have for a good assembly. That little purple bit over in the golden bit there, we've looked at that in detail. That is a prophage, so that's fine that it's done a bit of weirdness there because it's inserted itself in a funny place. And then we started looking at what's happening with our type 6 secretion system, because that's primarily why we wanted to get this full sequence. But then also we're going to do some other analysis later on as well. So what we found is that we've got that core type 6 secretion system gene clusters. Actually, it's divided into 2 clusters in different places on the chromosome, but that makes up that whole structure, the mechanism of the type 6 secretion system that sits in the membrane. What we didn't expect is that the type 6 secretion system has a duplicate. So cluster 1 at the bottom of the screen has a complete duplication at 100% identity at the top of the screen as well elsewhere on the chromosome. So we didn't anticipate this at all. We didn't see that when we had the contigs in the draft genome. It was only when we had this complete long read closed circular genome that we were able to find this there. So that was exciting for us and really justified doing the genome sequence there just on its own before we'd even got to do the VGRG sequences. Then we looked for the VGRGs, and we found that there's VGRGs here by the core cluster, which is good. So there tends to be one by the core cluster. So that's there to have one to load up onto the type 6 secretion system to inject into the competitor cells. But then we could find the others quite clearly elsewhere in the chromosome as well. So we've got a whole big bunch of them up at the top of the screen, and then one VGRG elsewhere on the chromosome as well. So now we can quite clearly tell how many there are, and we can investigate them in more detail around the chromosome. We also found that CONTIG2 has a VGRG. So this requires some more investigation from us. We're not quite sure whether this is going to be a self-replicating extrachromosomal circle or whether this is going to be an extrachromosomal circle that's transient, quite like the GGI. The gonococylogenic island can transiently come out of the chromosome and then go back in. Maybe this is something that, due to recombination between VGRGs, this is something that we've just captured as we've been doing the sequencing. We do know, when looking at other VGRGs in the chromosome, they can be present with repeat regions associated with them. So in that top bit of gene there, those red bars are repeat regions that may cause some recombination between them. We do see some VGRGs that are truncated. So in the middle set of genes there, you have the back half of the front half of VGRG gene, but not the end of the gene, and in the bottom sometimes you can see we've just got the end of the gene but not the beginning of the gene. So there seems to be lots of recombination happening with the VGRGs, and whether this is something that's as a result CONTIG2 or whether it's self-replicating, we're not entirely sure yet, so that requires some more investigation there. So long-read sequencing for bacteria, very much in favor of doing this. It has many, many benefits for us in being able to see the whole chromosome structure of the bacteria again. So in a space of about 20 years, we've come back to being able to see what's actually happening with the bacterial chromosome as a whole. It clarifies copy numbers for paralogous genes and for repetitive regions in the chromosome that are unclear from contig assemblies, from those short read contigs that we've relied on for so long and just being quick drafts. I didn't mention it here, but in the Neisseria, we have these Correa repeat enclosed elements. These are elements that are about 100, 150 bases, and they're present at 100 plus, more than 100 copies in the Neisseria chromosomes. So these really do complicate assemblies, particularly with shorter read sequencing. And also they can invert themselves. So that really complicates things as well. So they can be a big issue for assembly. They can be a big issue for trying to work out the structure of the chromosome. And they can be a big issue as far as what's going on dynamically within a chromosome as well. There are many paralogous sequences in a Neisseria genome. There's lots of genes that are present in different copy numbers, whether it's exactly identical or just really closely identical to one another. There's IS elements, and these things are true for lots of bacterial species, including the presence of prophage and various different permutations of prophage, whether they're intact or they're being degraded versions of protophages or just fragments of prophages that are still left around. So we can identify duplicated regions in the chromosome that are otherwise missed due to the nature of contig assembly. So there might be genomes out there in the databases that we have no idea even that there's these duplicated regions, the things that are going on in the chromosome. And until we do a complete genome sequence with a long-read technology to be able to do the complete assembly into a circle, we just aren't going to have any idea that those duplicated regions are in there. And so from this, we can identify dynamic and transient events, these creations of these little extra chromosomal elements formed during rearrangements. We might be able to pick those up by doing this sequencing, which otherwise would be lost in the mass of data from the draft genomes where they're just assembled into contigs, and capture things like these little flipped sequences that are present in short inversions. So some of these are present in the raw sequence data that maybe even for the long technologies will be lost out because they're not part of a larger consensus. But these short inversions, they're occurring dynamically in the bacterial chromosome, and they're important biologically for us to understand things like promoter regions and genes and short bits of the chromosome that are inverting, that are changing, that are slipping, and understanding biologically what that means for what's happening within a bacterial cell.